Hi everyone! This is the second of 3 videos that I'm making for the MCAT 2015 series. This video is going to focus on Foundational Concept 1B, and over here I've actually written out what Foundational Concept 1B is, and I will be talking about the transmission of genetic information from the gene of the protein. And what this foundational concept essentially is getting at is asking the question of how proteins are actually made. How are proteins made, right? So when we think about this question, there are 2 words that come to mind, and that's transcription, and translation. And these words should be familiar to anyone who's taken a high school biology course. They're very fundamental to biological systems. And in this video, I'm going to focus only on transcription and translation of eukaryotic cells. There are subtle differences between eukaryotic and prokaryotic transcription and translational processes. So with transcription, I like to think of the word transcribe. So in transcription, we are transcribe, which means to write. We're transcribing the gene into a readable format that will then make proteins. So we're transcribing DNA into RNA. RNA is the format that can be read by enzymes that need to make it into proteins then. So here I've drawn a chromosome, and in green here I've marked what a gene looks like. And when we take a closer look at a gene, you would see that, it is DNA, and DNA we know is double-stranded. And again, we're focusing only on eukaryotic cells, so here I'm drawing nucleotides. And what you have to know, and you probably already know, is that DNA is composed of 4 main nucleotides. Oops, that's the C. And here I've drawn them. A's pair up with T's, G's pair up with C's. Let's make that clear. A's pair up with T's, and G's pair up with C's. And the difference between DNA and RNA is that instead of having a T for RNA, we substitute that with a U. We also have to know that these nucleotides are categorized into 2 main groups. There are pyrimidines, and purines. And pyrimidines consist of cytosine, uracil, and thymine. So the C's, the T's, and the U's are pyrimidines, whereas the purines are adenine and guanine. Okay, and the way I think of it, or the way I remember it for exams, is we're using the mnemonic cut the pie. So pi meaning pyrimidine, see that? Yeah, that's cool. And then CUT goes with this CUT, okay? So a quick note about transcription before we go on to translation is that the actual process of transcribing DNA to RNA is done by an enzyme by the name of RNA polymerase. So the next step in the processes that lead to protein production is called translation, and that's when we, let's actually get blue here to stay consistent, when we translate the RNA that we made through transcription, which we've now labeled as mRNA because that's what it is actually. That stands for messenger RNA, so we translate the mRNA. There are many kinds of RNA, so it's good to distinguish what kind this is, and that's mRNA. So translate mRNA into amino acids, which, then essentially are the proteins. So here we have the mRNA, and it is capable of leaving the nucleus after transcription. And first, before it goes through translation, it goes through a series of what's known as post-transcriptional modifications. So let's actually talk a little bit about this. Modifications. There are three main ones that I'm going to highlight and not say too much about them, but the first one you should know is that the mRNA gets a poly-A tail, so a series of A's get added to one side of the mRNA. Poly-A tail. Oops. Butchered that. So that's a poly-A tail, and then there, on the other side you get a GTP, which is similar to ATP, but not quite. It has subtle differences. A GTP cap on the other side. And then one that we should be familiar with is that the introns of the mRNA, introns are removed. So if we take a closer look at what the mRNA is, you get the single-stranded mRNA, and some parts of this are actually introns, and some of them are exons. So, these parts could be exons, and these are exons. And the exons are the parts that code for the protein. Whereas the introns can get chopped off, and then the exons bind to each other. So you end up with something that looks like that. So taking a closer look at what exactly happens in translation, here we have the modified mRNA, and that's the one without the introns in it. It's just pure exons. And we have a ribosome, which sort of looks like a wonky mushroom, that can come and then bind to the mRNA. The ribosome is a protein that allows translation to occur. What it does is it takes amino acids from the neighboring regions, from just the cytoplasm floating around, and pairs up amino acids with 3 of the nucleotides found in the mRNA. So every 3 nucleotides in the mRNA is known as a codon, and every codon codes for 1 amino acid. And there's actually a really nice table that tells you which amino acid a group of 3 nucleotides would code for. So here is the ribosome, and that goes and binds to the mRNA. And it goes through these 3 steps. So the first step is the binding of the ribosome to the mRNA. The second step is its elongation. So the elongation of the amino acids that then form by the translation of each codon. So at the end, after 1 codon is read, you get 1 amino acid added. And then the second codon's read. So let's just say that's read next. And then the second amino acid's added, and then a third codon is read. And that's the third one that's added. So every time we read 3 of these nucleotides, we get another amino acid added to the end. And so you could just think of it like balls in a string elongating each time. And the final step, of course, is when you reach a special kind of codon known as a stop codon. So let's just say that's the stop codon. And when the ribosome gets to the stop codon, it tells the ribosome that it needs to dissociate. So the ribosome lets go of this mRNA, and that goes on its way. And this actually does not code for an amino acid, it just ends the string here. So these are bonded by peptide bonds, and this is actually, let me erase that so that you don't confuse that for a bond, but that's where it ends. And so those are the 3 main steps of translation. And there you have it. That's transcription and translation.